So today's topic is fertilization and gametes formation in plants. So before starting this particular topic, we need to know the structure of the flower or the parts of a flower which is involved in reproduction of plants. So this is the structure, basic structure of the flower. The outermost part is called sepal. The colorful parts are called petals. Then this part is called anther. The long part is called filament. This long part again from the female reproductive organ is called style. This uppermost part is called stigma. This round shape is called ovary. Green colored part, this part is the egg. This is the egg. Then this blue colored part, this is the ovule. Okay, later on ovaries develops into fruits and ovule develops into seeds. Now a group of petals are called corolla. That means collection of petals together will be called corolla. All the petals in the flower will be called corolla. A group of sepals together, many sepals will be called calyx. That means a group of sepals is called calyx, or calyx is the collection of all the sepals present in the flower. Now see, anther, filament. Anther plus filament together they will make the male reproductive part of the flower, which we call stamen. Okay, what is stamen now? Stamen is the male reproductive part of the flower which is composed of anther plus filament. This anther and filament together they will make stamen. Similarly, ovary plus style plus stigma. Together, these three will make the female reproductive part of the flower, which we call carpel. Now, what is carpel? Carpel is the female reproductive part of the flower, which consists of ovary, style, and stigma. Anther is very important because anther, this portion, will contain the pollen grains or the male gametes. Where the male gametes will be present, they will be present inside the anther here. Okay. Again, the stigma, this part is also very important because the pollen grains from anther will go and fall upon stigma, and then they will enter through this part and then they will reach the egg in order to fertilize the egg. So our next topics will be megasporogenesis and microsporogenesis. Megasporogenesis is the formation of female gametes, that is the egg, in plants, and microsporogenesis is the formation of male gametes, that is the pollen grain in case of plants. Let's first start the megasporogenesis. So here will be a tissue which will be called nucellus tissue. These tissues will have cells which will be called MMC, that is MMC, which is called megaspore mother cell. These megaspore mother cells will later form the megaspore, and then that megaspore will divide and differentiate into egg. These MMCs, they will do meiosis, meiotic division. So when they will be doing meiosis, so at first, okay, remember these MMCs are normal somatic cells of the plant body, which will be 2N. So these 2N, when they will do meiosis, they will form NN. I hope you know this. Meiosis is a reductional division. As a result of meiosis 1, 2N will get, okay, 2 cells will get consisting of NN. Then meiosis will be completed once meiosis 2 is completed. So as a result of meiosis 2, we will again get 4N, that is we'll get a tetrad. Now the arrangement will be like this only. Each one of these will be called one megaspore. So here we will have 4 megaspores that we have obtained from the megaspore mother cell. that is MMC, as a result of meiosis. Meiosis 1 will give us 2N and meiosis 2 will give us 4 cells consisting of N. Each of them will be called megaspore. Remember, the upper portion, this end is called chelagel end and the lower end is called micropyle end. Chelagel end and micropyle end. Now here is a twist. Not all the 4 megaspores will participate in the further process. Only one megaspore present in the chelagel end, this end, will participate in the further process, and the remaining 3 megaspores will degenerate. They will not participate in the further process. Now let's start our story from this point. Okay. Now this surviving megaspore from the chelagel end will continue the process. This particular megaspore, what it will do? It will do mitotic division. Okay. But remember, as a result of mitotic division, only karyokinesis will be there. That means only nucleus will divide, not the cells. Anyway, when mitosis will be done, then one nucleus will divide into 2. Okay, this is the first mitotic division done by the megaspore, surviving megaspore. Then again another mitotic division will be done, which will be the second mitotic division. Again karyokinesis will be there, not cell division, only nuclear division. So 2 nuclei will become 4 now. Again third mitotic division will be there. As a result of this third mitotic division, again karyokinesis will be there. 4 nuclei will now become 8. 1, 2, 3, 4, 5, 6, 7, 8. Now here mitotic divisions will be completed. Total 3 mitotic divisions will be done. As a result of that, 8 nuclei will be obtained. Now after karyokinesis, that means after nuclear division, cell division will be there. Now cytokinesis will happen. Cytokinesis means actual cell division. Now see what we will obtain. Here cells will be formed. That means plasma membrane will surround the upper 3 nuclei. Then here also cells will be formed. Plasma membrane will surround the lower 3 nuclei, but the remaining 2 nuclei present in the middle, the central nuclei, they will not be covered with any plasma membrane. They will not be a part of any cell. Okay, these 2 nuclei will be called polar nuclei. They are naked. They are not covered by any cell layer. The upper 3 cells will be called antipodal cells. The lower, this cell and this cell, these 2 cells will be called synergids. These 2 nuclei will be called polar nuclei. Polar nuclei. And the middle, this bigger cell, this is our egg, which is the plant gamete. Okay, female gamete for the plants. Now this particular stage is called 7 cell 8 nuclei stage, because here we have 7 cells. One, the bigger one, then 2, 3, 4, 5, 6, 7 cell. If you count the nuclei, we'll have 1, 2, 3, 4, 5, 6, 7, 8. 7 cell 8 nuclei stage will be this one. As a whole, this particular stage is called the embryo sac. Okay, this is the embryo sac. This was all about megasporogenesis. Now let's start microsporogenesis. See, if we cut the anther and if you observe it from the top view, anther will look like this. Okay, here these four structures are called megasporangium. These are like pouch, these are like bag. inside megasporangium will be present many MMC. Again MMC. This time it's not called megaspore mother cell. This time this will be called microspore mother cell. Microspore mother cell. Okay. Later on, these MMCs will become microspores and then pollen grains. Now here let's focus on only one MMC. Okay. Each MMC will do the same thing, but here we are only focusing on one MMC. This MMC, first it will do meiosis 1. Remember again, MMCs are somatic cells. They are 2n, twice n. This MMC, 2n MMC will do meiosis 1 and meiosis 2. So after meiosis 1, we'll get this type of thing. And then after completion of meiosis 2, we'll get again tetrad. N. These are called microspores. Okay. Unlike the megasporogenesis, here the arrangement was linear. Okay. All the megaspores were arranged in a linear structure like this. The tetrads was a linear tetrad. But this time it will be circular. The arrangement is somewhat like this. Okay, like this. Another difference is that here only one megaspore was participating in the further process. Only one survived from the chelagel end. But here all the 4 microspores, all the microspores will participate in the process, not only one. Okay. Here there is no such concepts like chelagel end and micropyle end. So each one of them will participate in the process, but here again we are taking example of only one. Remember, all of them will participate. But it's not possible to study all the 4. So here we are only taking one microspore out and then we'll study the further process. This microspore, that means all of the microspores, what it will do? It will do one mitotic division. But before that, what it will do, see, it will form 2 layers. Okay. One is called exine. Okay. This outer layer, which is this continuous layer, this part is called exine. And the inner part, inner layer is called intine. Exine and intine will be formed around the microspore. After that, division will be there. What type of division? Mitotic division. As a result of this mitotic division, what will happen? We'll get 2 cells. One bigger cell and the another one will be the smaller cell. Okay, then again intine and exine will be present. I am drawing it roughly this time. This bigger cell will be called the vegetative cell. And the smaller cell will be called the generative cell. The bigger cell, that is the vegetative cell, will form the pollen tube. The generative cell will become the pollen grain, which is the male gamete. Now, what is this pollen tube? I hope you already know about pollen grain. This will fertilize with the egg later on. But what is the role of this pollen tube? The pollen tube will make a tube like this. The pollen tube is a tube like this through which the pollen grain can enter and reach the egg. One more thing which is important here is the role of these antipodal cells and synergids. Antipodal cells here, these upper 3 cells, they will provide nutrition to the egg. And synergids, what they will do, they will guide the pollen grain coming through the pollen tube to the egg. They will show the route, or they will guide the male gamete to reach and fertilize the female gamete, that is the egg. These are the roles of antipodal and synergids. Now the last part, that is the fertilization part. See what will happen. This is the stigma of the female. This will be the ovary, and here inside the ovary, synergids, eggs, antipodal cells, and the entire embryo sac is now present here. Okay. Now, when the pollen grain will reach this particular portion, this will be the pollen tube, okay, which is formed from the vegetative cell. This pollen tube, the pollen grain will enter the female reproductive organ through the pollen tube. Remember one thing, this pollen grain during its journey through the pollen tube will again divide. Okay, a last mitotic division will be done. So first mitotic division was done in order to make the vegetative and generative cells, 2 cells, and the second mitotic division will be done during the journey through the pollen tube to the egg. Okay, so now we have 2 pollen grains, N. Okay, because I hope you already know the result of mitosis is equational. Whatever will be present in the parent cell will be in the daughter cells. Okay, if it is N, then NN. If it is 2N, then the result of mitosis will be 2N. Similarly, meiosis 2 is also same, and the result of meiosis 1 is NN. Okay, I hope this concept is clear. If it is not clear, then I can make a separate video to make you understand meiosis and mitosis. I am assuming that you already know. Anyway, these 2 NN, that means the 2 pollen grains which we got as the result of the last mitotic division, these 2 will move through the pollen tube like this, and then they will, one of the pollen grain will fuse with the egg. Egg was already N. Another pollen grain will fuse with the egg, then now together they will become 2N, and this will become the zygote. Okay, 1N from the pollen grain, 1N from the egg, together they will become the zygote 2N, and this will later become the embryo. Okay, what about these 2 polar nuclei? See, we had another pollen grain, right? This second pollen grain will fuse with this polar nuclei, and together they will become 3N. Why 3N? Because already there were 2 polar nuclei, N. Another pollen grain fused with them, so it's 3N. This 3N, this will later become a tissue which is called endosperm. This endosperm, this will provide nutrition to the growing embryo. This egg and pollen grain together, they will make the embryo, right? This embryo will grow eventually, and that embryo will require some nutrition which will be provided by the endosperm. Example of endosperm is coconut water, and the white portion of the popcorn that pops out, that is also example of endosperm. Okay, so this particular stage is now called triple fusion. This part is called triple fusion because here 3N is formed, and another term is there which is called double fertilization. Why double fertilization? Because fertilization is doubled here. One fertilization is this one, another fertilization is this one. So this is called double fertilization, triple fusion in flowering plants. I hope this simple explanation was helpful enough. If you have any kind of doubt, then you can comment down below. I hope the video was helpful enough. If you want to take detailed coaching of slet or neet, then you can contact in the given number, or you can comment down below. I used to provide detailed coaching of slet and neet under life science category, as well as I used to provide guidance for your paper 1. So quickly contact and make your dreams into reality. Bye and all the best.